Hi, welcome to Care ALS Plus Capsule.
Hi, everyone.
Today, we're diving into something pretty fundamental,
Yeah.
neurodegeneration.
Hmm.
It's basically like uh, trying to understand what happens
Right.
when some of the most critical cells in our body, our nerve cells, start to kind of go awry.
Yeah, absolutely.
And you shared some really, really interesting material with
I
us.
did.
And our goal today is to kind of pull out the key ideas so you can grasp this important topic
Right.
without getting lost in all the, the complicated details.
Yeah.
This is for you, the learner,
Yeah.
who wants to understand the basics and maybe even share that understanding
Exactly.
with others.
What we're exploring today really touches on the core of so many brain-related conditions.
Okay.
Your sources highlighted things like protein clumping,
Yeah.
communication breakdowns between brain cells,
Yeah.
problems with how cells transport things internally, and even some futuristic ideas about how tiny particles could help.
Mm-hmm.
We want to kind of weave these together so you see the big picture
Yeah.
and understand why this area of research is so vital. So let's start right at the beginning.
Okay. Yeah.
What exactly is neurodegeneration?
Okay. So neurodegeneration in a nutshell.
Yeah.
Like, what's the main idea?
At its heart, neurodegeneration is the progressive loss of neurons.
Okay.
These are the nerve cells in your brain and also extending throughout your body.
Okay.
This loss can affect their structure, how they function, or both.
Yeah.
And it's a central feature of many disorders that impact the brain.
Okay.
While we often picture neurons just in the brain, remember they're everywhere,
Right.
controlling muscles, senses, pretty much everything. Interestingly, while we create loads of new neurons when we're young,
Oh, yeah. Heard of that.
that production slows way down in adulthood.
Really?
It's like our brain's initial investment has to last us a long time.
Wow. So if we're losing these irreplaceable cells,
Right.
that sounds like the root of a lot of serious issues.
It can be.
What are some of the conditions that fall under this, umbrella
Yeah.
of neurodegenerative disorders?
There are quite a few, and you've likely heard of some of them.
Yeah.
Alzheimer's disease and Parkinson's disease are two of the most common.
Yeah.
Then you have amyotrophic lateral sclerosis, or ALS, which you might know as Lou Gehrig's disease.
Right.
Other examples include prion diseases, Huntington's disease, spinal muscular atrophy, and spinocerebellar ataxia. What's striking is the sheer number of people affected globally.
Wow.
And while getting older is a major risk factor for many of these,
Okay.
our genes and the environment we live in also play significant roles.
So it's not just age.
Right.
There's other factors.
And it's also important to realize that a single neurodegenerative disease can have multiple things going wrong at the cellular level.
Okay.
It's rarely just one simple failure.
It's like a complex system
It is.
where if one part starts to fail, it can have kind of a ripple effect. Now, one of the key things that came up in the material was this idea of proteins clumping together. Why is that such a big deal in neurodegeneration?
Think of it like this. Proteins are the workhorses of our cells. And they need to fold into very specific shapes to do their jobs correctly.
Right.
In many neurodegenerative diseases, these proteins misfold and start to stick together, forming clumps or aggregates.
Okay.
These clumps can disrupt normal cell function and even spread the damage to neighboring cells.
Mm-hmm.
The rapid neuron loss seen in prion diseases really highlights how potent this protein aggregation can be
Oh, wow.
as a driving force in neurodegeneration. It's such a key feature that scientists even use the term prion-like
Oh, interesting.
to describe how this kind of spread might occur in other conditions like Alzheimer's and Parkinson's.
Okay.
It's as if these misfolded proteins can act as seeds, causing other healthy proteins to also misfold and aggregate.
So it's like a chain reaction almost.
It is.
Prion-like spread, that's a very vivid way to picture it.
Yeah.
And the sources mentioned some ways to actually detect these rogue proteins.
Yes, and this is a really exciting area of progress.
Yeah.
Techniques like RTQIC,
Right.
think of it like a super sensitive protein seed detection test,
Okay.
and PMCA,
Okay.
which essentially amplifies tiny amounts of these misfolded proteins so they can be detected,
Gotcha.
are being developed to find these disease-causing protein forms in cerebrotinal fluid, the liquid surrounding the brain and spinal cord,
Okay.
for prion protein and alpha-synuclein, a key protein in Parkinson's.
Right.
These tests have shown remarkable accuracy in research and are moving closer to becoming standard diagnostic tools.
That's great.
This could allow for earlier diagnosis and potentially earlier intervention.
That would be amazing.
It's worth remembering, though, that not all neurodegenerative diseases involve this kind of protein clumping.
Okay.
as their primary driver. There are other mechanisms at play in some conditions.
So while protein clumping is a major problem,
Right.
in many of these diseases, it's not the whole story.
Right.
Another thing that kept appearing was this idea of synaptic dysfunction.
Yeah.
What are synapses and why is their malfunction so important in neurodegeneration?
Imagine your brain as a giant network of computers,
Okay.
and neurons are the individual computers. Synapses are the connections between these computers,
Sure.
where they send signals to each other.
Right.
What's really fascinating and important is that in many neurodegenerative diseases, these connections start to fail before the actual neurons die.
Wow.
This happens in Alzheimer's, Parkinson's, Huntington's, frontotemporal dementia, ALS, and even after a stroke.
Oh, wow.
It's like the communication lines are going down even before the computers themselves shut off.
So the cells are still there,
Right.
but they're not talking to each other effectively.
Right.
That sounds like it would have a big impact.
It does, absolutely. In Alzheimer's, for example, the loss of these synaptic connections correlates much more closely with memory loss than the actual number of neurons that have died.
Okay.
Sometimes, in early stages, you might even see increased activity in some brain regions trying to compensate for these failing connections.
So they're working extra hard.
They are. They're trying to make up for it.
Mm.
In Parkinson's, the connections between neurons that produce dopamine start to degrade even before the dopamine neurons themselves are lost.
Oh.
In fact, the dysfunction at these synapses is often more pronounced than the cell loss, especially when symptoms first appear.
Right.
We see similar patterns in Huntington's and ALS.
Okay.
The communication pathways weaken before the cells themselves are gone.
So these synaptic problems seem to be a really early
Right.
and critical event in many neurodegenerative diseases.
Absolutely.
What's causing these communication breakdowns? Like, what's going on?
Well, those same misbehaving proteins we talked about earlier play a big role. Mutations in proteins like amyloid precursor protein, APP, in Alzheimer's,
Okay.
tau and alpha-synuclein in Parkinson's,
Right.
and Huntington in Huntington's have all been linked to synaptic problems. These faulty proteins can interfere with the normal machinery at the synapse,
Okay.
disrupting the delicate balance needed for proper communication.
And the sources also mentioned a cellular cleaning system.
Yeah.
That can get overwhelmed.
Yes. That's the autophagy lysosome pathway, or ALP. Think of it as the cell's recycling and waste disposal system.
Okay.
It's crucial for keeping the synapse clean and functioning correctly by getting rid of old or damaged proteins.
Gotcha.
However, when those aggregating proteins build up, they can clog up or damage the ALP at the synapses.
Oh, no.
Making the dysfunction even worse. It's like the garbage trucks are on strike and the trash just keeps piling up, interfering with everything.
It's a good analogy. Okay, so we have protein clumps
Right.
and communication breakdowns. What about these axonal transport defects?
Yeah.
Axons are those long fibers of nerve cells, right?
Exactly. The axon is like the long cable that transmits signals away from the neuron's main body. Axonal transport is the process of moving essential supplies, things like mitochondria for energy and the building blocks for neurotransmitters,
Okay.
along this cable between the cell body and the distant synapse. In neurodegeneration, this internal delivery system starts to falter.
Right.
It's like a railway system where the trains are delayed or not reaching their destination.
Gotcha.
This can lead to the synapse not getting what it needs to function,
Right.
and the neuron can start to degenerate, either from the synapse back towards the cell body that's dying back,
Okay.
or sometimes in other patterns.
So it's a bit like if the limbs of a tree aren't getting nutrients from the roots.
That's a great analogy.
Yeah.
And your sources highlighted how genetic mutations can underscore the importance of this transport system.
Okay.
For instance, mutations in the NEFL gene are linked to Charcot-Marie-Tooth disease,
Yeah.
a peripheral neuropathy. And mutations in MAPT, which codes for the tau protein involved in Alzheimer's and other tauopathies, also affect axonal transport. These show how critical a healthy axon and efficient transport are for neuron survival and function.
Wow. We're seeing problems at so many levels.
Yeah.
Now, the outline also mentioned transcription issues. That sounds like it gets down to the cell's kind of fundamental instructions.
It does. Transcription is the process where the DNA code in a cell's nucleus is read to create RNA,
Okay.
which then serves as a template for making proteins. Your sources pointed out that problems with DNA repair mechanisms,
Okay.
specifically something called transcription-coupled nucleotide excision repair, or TCNER, can contribute to neurodegeneration.
Wow, that's a mouthful.
It is. TCNER. Think of TCNER as a spell checker for the DNA when it's being read to make RNA. If the spell checker isn't working properly, errors can accumulate.
And these errors can then lead to problems with the proteins being made.
Exactly. Mutations in a gene called senet-taxin, or SETTX, which helps manage the interactions between RNA and DNA, have been linked to certain forms of ataxia and ALS.
Okay.
This suggests that unusual structures called R-loops, which are temporary tangles of RNA and DNA that can form during transcription,
Right.
might become problematic and contribute to neuronal death when the repair mechanisms are faulty.
It's incredible how many, intricate processes need to work perfectly
It is.
for these cells to stay healthy.
Yeah.
The outline also mentioned that it's not just the neurons themselves that are involved. There are non-cell autonomous mechanisms and the role of glial cells.
Yeah.
What are they up to?
This is a crucial piece. Neurons don't exist in isolation. They're surrounded and supported by other brain cells called glial cells, like astrocytes and microglia.
Okay.
Astrocytes are like the support staff for neurons,
Right.
providing nutrients and helping to regulate their environment. Microglia are the brain's immune cells, cleaning up debris and fighting off infections. In neurodegeneration, these glial cells become activated, a process called reactive gliosis.
Okay.
While initially this might be a protective response, in the long run it can sometimes become harmful, contributing to inflammation and further damage.
Well,
Interestingly, in some neurodegenerative diseases, the glial cells themselves can also become targets of those misfolded proteins we discussed earlier.
so they're not immune.
No, they're not. What's really emerging is the understanding that there's a complex communication network between neurons and these glial cells.
Okay.
And disruptions in this communication play a significant role in the progression of these diseases. Researchers are even using brain imaging and looking at biomarkers in fluid to try and understand and track the contributions of these glial cells.
So it's not just the neurons getting sick. It's like the whole neighborhood is affected.
It is.
That makes sense. Now, thinking about all these things that can go wrong,
Right.
why are neurons seemingly so vulnerable? What makes them so susceptible to these issues?
Several reasons contribute to their vulnerability. First, as we mentioned earlier, neurons have a very limited ability to regenerate once they're lost.
Okay.
Second, they have incredibly high energy demands to support all their electrical activity.
Right.
And this high energy use leads to the production of harmful byproducts called reactive oxygen species. Third, their long axons and dendrites require a really efficient transport system over considerable distances, which is prone to disruption.
Yeah.
And finally, they are heavily reliant on the support of those glial cells for their survival and function.
So it's like a combination of factors.
It is.
It's a combination of being highly specialized, long-lived, and dependent on a complex support system that makes them susceptible to these various insults. It paints a picture of cells living life on a tightrope.
It does.
So when all these problems accumulate, how do the neurons actually die? What are the different ways this can happen?
Neuronal death, which we can sometimes see on brain scans,
Okay.
and by detecting proteins released from damaged neurons, can occur through various mechanisms. There's apoptosis, which is a programmed, more controlled form of cell death. Then there's necrosis, which is often a more rapid and uncontrolled death. Other mechanisms include necroptosis, ferroptosis, phagoptosis, autophagic cell death, and pyroptosis, each with its own distinct molecular pathways.
Wow.
For example, if the powerhouses of the cell, the mitochondria, are damaged,
Right.
they can trigger cell death either directly or by releasing certain proteins. Similarly, the lysosomes, those enzyme-filled sacs,
Okay.
can become leaky due to protein buildup, and the release of their digestive enzymes can lead to the neuron's demise. It's a complex picture, and often multiple of these mechanisms might be happening simultaneously.
It sounds like there isn't just one way for a neuron to die.
There isn't?
There are many different paths. And it seems like all these different hallmarks we've discussed, the protein issues, the synapse problems, the transport breakdowns,
Right.
they're all interconnected.
Absolutely. They don't operate in silos. They're all part of a complex web of interactions. For example, problems with synaptic function can lead to an overstimulation of neurons, called excitotoxicity, which in turn can stress the energy production of the cell and increase oxidative stress.
Wow.
Similarly, the buildup of misfolded proteins can disrupt the the cell's energy balance,
Okay.
interfere with synaptic function, and ultimately contribute to neuronal death. This interconnectedness is a key challenge for developing effective treatments, because targeting just one aspect might not be enough to halt the overall disease progression. It really highlights the potential need for therapies that can address multiple issues at once.
That makes sense. Trying to fix one broken part
Right.
in a very complicated machine
Right.
might not be enough. So, with all this understanding of what goes wrong,
Yeah.
what are the current treatments for major neurodegenerative diseases like Alzheimer's, Parkinson's, and ALS,
Yeah.
and what are their limitations?
This is a really important area to discuss.
Yeah.
While we've made progress, the current treatments for these major neurodegenerative diseases primarily focus on managing the symptoms rather than fundamentally changing the course of the illness. Take Alzheimer's, for instance. Aducanumab is a newer antibody treatment that can help clear amyloid plaques from the brain.
Okay.
But its effect on cognitive decline is still debated.
Right.
We also have older drugs like cholinesterase inhibitors that can temporarily improve memory and thinking in some people, but they don't stop the underlying disease.
Okay.
Similarly, memantine can help with some of the later stage symptoms.
So these are helping manage the effects,
They are.
but not really tackling the root
Right.
cause.
Precisely. In Parkinson's disease, the main approach is to replace the lost dopamine in the brain using medications like levodopa or dopamine agonists. These can be very effective at managing motor symptoms, but they don't halt the progression of the disease and can have side effects.
Okay.
For ALS, we have a few medications like riluzole and edarvone that have shown modest benefits in slowing the disease's progression or prolonging survival.
Okay.
But again, they are not cures.
Right.
A major hurdle in developing more effective treatments for all of these conditions is the blood-brain barrier, a natural defense system that makes it very difficult for many drugs to get from the bloodstream into the brain where they're needed.
So the medicine can't get to where it needs to go.
It can be very challenging.
Oh, wow.
It's like trying to deliver medicine to a heavily guarded fortress.
That makes it much harder.
It does.
The blood-brain barrier sounds like a significant obstacle.
Yeah.
That's where the idea of using nanotechnology comes in, right?
It is.
These tiny particles that might be able to bypass this barrier.
Exactly. Nanoparticles, which are incredibly small, we're talking about the scale of a billionth of a meter, are being explored as a way to deliver drugs.
Wow.
And even genes directly to the brain,
Wow.
potentially crossing the blood-brain barrier more effectively.
That would be huge.
Researchers are using a variety of materials to build these nanoparticles, including natural and synthetic polymers, as well as inorganic materials like gold, silver, and cerium. The potential advantages are huge. They could carry large amounts of drugs, have low toxicity, improve drug delivery to the specific areas that need it, and protect the drugs from being broken down too quickly by the body.
So how do these tiny particles actually get across this barrier?
Right.
Does it just sneak through unnoticed?
It's a bit more sophisticated than that. Their ability to cross depends on several factors, including their size,
Okay.
what their surface is coated with, their surface chemistry,
Okay.
and their electrical charge.
Gotcha.
By carefully designing these properties, scientists can make them more likely to cross. For example, coating them with certain molecules can help them interact with the cells of the blood-brain barrier in a way that allows them to pass through,
Okay.
or even evade the pumps that normally push foreign substances back out of the brain. Liposomes, which are tiny spheres made of fat-like molecules, and polymeric nanoparticles are particularly well studied for this because their surfaces can be easily modified to target specific molecules or cell types in the brain.
That sounds really innovative.
It is.
Could you give us some specific examples of how these nanoparticles are being used in research?
Yes.
You mentioned gold, silver, and cerium.
Certainly. Gold nanoparticles, or AUNPs, have been used for both imaging the brain and for delivering drugs in a targeted way. They're good for imaging because they interact with X-rays differently than surrounding tissue, providing better contrast. They can also be coated with molecules that specifically bind to disease-related targets in the brain, like the amyloid plaques in Alzheimer's, potentially helping to break them down.
Oh, wow.
Silver nanoparticles, or AGNPs, have been shown to reach specific brain regions like the hippocampus after injection and have been used to deliver drugs for conditions like brain tumors.
Okay.
Some research also suggests they might have anti-inflammatory and antioxidant properties. However, there are also concerns about their potential toxicity at higher concentrations. Cerium oxide nanoparticles are interesting because they can act as antioxidants, reducing the harmful reactive oxygen species that contribute to neuronal damage. They've shown promise in animal models of Alzheimer's by reducing amyloid and protecting neurons, and in models of stroke and ALS by improving motor function. Some studies even suggest they might help promote the growth of new neurons.
That's amazing. What about those organic nanoparticles like liposomes?
Organic nanoparticles like liposomes, solid lipid nanoparticles, and PLGA nanoparticles are attractive because they're generally biocompatible and can protect the drugs they carry from degradation. Liposomes, being similar to the cell membranes themselves, can sometimes fuse with the blood-brain barrier, releasing their cargo inside. They can also be modified with targeting molecules to specifically bind to certain cells or proteins in the brain. PLGA nanoparticles offer the advantage of controlled drug release over time, and can also be engineered to have an affinity for neurons and even travel backwards along the axon, potentially delivering therapies to the main cell body.
It sounds like this field is really pushing the boundaries of how we can treat these diseases.
Mhm.
Are any of these nanoparticle-based therapies actually being used in patients right now?
That's where things are still developing. While there's a lot of excitement in the research lab, we're still in the early stages of seeing these translate into widespread clinical use for neurodegenerative diseases. There are a handful of nanoparticle-based formulations in clinical trials for conditions like Alzheimer's and Parkinson's, and one lipid nanoparticle formulation has been approved for a different condition called transthyretin-mediated amyloidosis,
Okay.
showing that this approach can be successful.
Right.
Other ongoing trials are exploring things like gene therapy delivered via nanoparticles for conditions like Huntington's.
So we're seeing the first steps towards using these tiny tools in the clinic. What are some of the big hurdles that still need to be overcome?
There are several important challenges. Safety is always the top priority. We need to ensure that these nanoparticles are not toxic to the brain or other parts of the body.
Right.
Manufacturing them consistently and at a large scale is another hurdle. And then there's the regulatory pathway. Getting these novel therapies approved for use in humans requires rigorous testing and evaluation.
Yeah, that makes sense.
However, the potential benefits are enormous. And the hope is that these nanotherapeutics will eventually allow us to target the underlying causes of neurodegenerative diseases and potentially even reverse some of the damage rather than just managing symptoms.
This has been an incredibly insightful look into the world of neurodegeneration.
Yeah,
From the fundamental processes that go wrong
right.
to the cutting-edge research using nanotechnology to find new treatments.
Yeah,
So to summarize, we've explored the progressive loss of nerve cells,
right.
the roles of misfolded proteins,
Yeah.
the breakdown of communication between neurons, problems with cellular transport, and the exciting potential of nanoparticles to overcome the challenges of treating these complex conditions.
Yes, and thinking about all the interconnected mechanisms we've discussed, it really makes you wonder. Could future treatments involve personalized combinations of these nanotherapies, each precisely targeting a different aspect of the disease in an individual,
Oh, wow.
instead of a one-size-fits-all approach? Could we see highly tailored treatments that address the specific molecular issues at play in each person's neurodegenerative condition? It's a fascinating and rapidly evolving field.
Absolutely. We hope this deep dive has given you a solid foundation for understanding neurodegeneration and the innovative research underway. It's a complex but critically important area. And the more we understand it, the better equipped we are to support those affected and contribute to finding solutions. Thanks for joining us on Care ALS Plus Capsule. We'll catch you next time.